So now it's my great pleasure to introduce our keynote speakers, Dr. Rhonda Kost and Dr. Jonathan Tobin. Dr. Kost is Director of the Clinical Research Support Office and Co-Director of Community Engaged Research Core of the Rockefeller University Center for Clinical Translational Science in New York City. She's also the Vice Chair of the Rockefeller Institutional Review Board and Associate Professor of Clinical Investigation at Rockefeller. Dr. Kost is a physician specializing in infectious diseases and was a medical staff fellow in the section of virology in the NIH's National Institute for Allergy and Infectious Diseases. She co-led the development of the Rockefeller Community Engaged Research Navigation Model, which is an approach to foster sustainable research partnerships between basic scientists and affected communities, and to align the aims of community engaged and mechanistic researchers to design and conduct full spectrum translational research with meaningful outcomes for all collaborators. She's the PI for Empowering the Participant Voice, which is a multi-site study funded by NCATS to streamline adoption of the Research Participant Perception Survey, which is a validated set of participant-centered measures of the research experience. She was a co-author of the 2018 National Academy of Sciences report that was titled Returning Individual Research Results to Participants, a Guide for New Research Paradigm. Dr. Jonathan Tobin is a cardiovascular epidemiologist and president and CEO of the Clinical Directors Network, a New York City-based practice-based research network, an AHRQ designated Center of Excellence for Practice-Based Research and Learning. The CDN is a network of safety net PBRNs dedicated to improving clinical outcomes for low-income and medically underserved communities by creating community academic partnerships around research, education, training, and service. Under Dr. Tobin's leadership at CDN, over 600 federally qualified health centers, 1,000 physicians, and nearly 400,000 patients have been recruited into clinical trials and observational studies. Dr. Tobin is also the co-director, along with Dr. Kost, of the Community Engaged Research Core at the Rockefeller University Center for Clinical and Translational Science, as well as a senior epidemiologist and adjunct professor at the center. He's also a professor in the departments of epidemiology and population health at Albert Einstein College of Medicine and the Montefiore Medical Center in the Bronx. Welcome, Drs. Kost and Tobin. We're looking forward to learning from you about how to embed community voice and involvement at all levels of the translational research spectrum.
All right, good morning. Thank you, Lee. Thank you for that introduction. We're quite honored to be included in your community research exchange, and thank you for the invitation and the opportunity to share our 15-year experience together building full spectrum translational research teams. So I'll start first with a description of our academic home, my academic home, Rockefeller University. We're somewhat unusual among academic medical centers, academic research institutions. We're not a medical center. We have no departments. We have no natural patient population. We're organized around 60, 70 individual labs led by PhD and MD PhD heads of lab. And we have a more than 100-year tradition of translational research, though typically early on the spectrum. In fact, our tagline, Science for the Benefit of Humanity, and our many Nobel Prizes speak to our commitment to translation, but we are on the left end of the spectrum. We have a small research-only hospital. We're AHRP accredited, and the small portfolio of protocols that we conduct every year are mostly investigator-initiated, mostly mechanism of disease or early phase clinical trial, although in the last decade, we've added community-based participatory research, sort of oscillating on the two ends of the translational spectrum. With our Clinical Translational Science Award in 2006 and a specific charge to conduct community-engaged research, we knew immediately that we would need partners. Being a mechanistic scientific research institute, we would need a community partner, and joined with Dr. Tobin and Clinical Directors Network from the outset to develop a model that would allow us to build on our strength as a university with great strength in laboratory-based science. Next slide, please. So I've heard the translational research spectrum mentioned several times in the introductory remarks, and just to make sure we're all talking about the same thing, sometimes we see T1 through T4. where the most basic research is left off the spectrum, but particularly at Rockefeller, we think about T0 through T4. And historically, research has fallen into one of these phases, these silos, although, and the charge of the clinical translational science is to accelerate moving across this spectrum. And we believe that actually, rather than conducting research in parallel or within these silos, the key is to integrate the design and conduct, the way we think about research across the spectrum. Next slide, please. So to accelerate moving discoveries from the lab to the bedside, from the bedside to the curbside, to policy and back, and integrating research across these silos requires rethinking how to design full spectrum translational research and how to form the sustainable multi-stakeholder community engaged partnerships essential to designing full spectrum translational research. Next slide, please. I realize I'm preaching to the choir from the introductory remarks, and I just still need to make foundational comments that the core principles of community engagement are essential, but not, of course, not sufficient. And as I was preparing this and looking at these set of principles from a decade ago, it feels like they're speaking to investigators about learning about the community, and yet they apply in all directions for all stakeholders to be humble in learning about the other communities, the other stakeholders, to build trust and seek commitment, to respect self-determination, and to recognize that partnerships require time, must build on everyone's strengths, and require releasing control on all sides, sharing of governance and resources, and long-term and sustained commitment. That the partnership can't begin and end with the project. It needs to start long before the project, be sustained between projects, and live on beyond the end of any particular grant. Next slide, please. A couple of weeks ago, we were at the Translational Science meetings in Chicago, and Dr. Phil Alberti from AAMC Center for Health Justice gave a lovely closing plenary about their efforts and their work in articulating principles of trustworthiness, sort of flipping this, why don't communities trust us? How can we get communities to trust us? Flipping that to how do we become trustworthy? And I refer you to their website where the principles and toolkits and narratives really take the principles of community engagement to a more sophisticated level, addressing the humility and listening and demonstration of commitment that's necessary to truly engage and address health justice. Next slide, please. I was also struck by a slide from his presentation where through a survey of several thousand individuals in the community asking about the trustworthiness of different sectors of the community, you'll notice that universities are at the bottom. I'm sure this continuum goes further down. We're not at the very bottom, but it's humbling to see and shows that we have our work still cut out for us. Next slide, please. So how do T1, T2 scientists and communities engage? By practicing broadly engaged team science, coming from a very scientific institution. I often see, at least a decade ago, 5 years ago, when we ask investigators to describe their scientific team or who the stakeholders are in their team, they're often identifying their other scientific collaborators, their peers in the lab next door or at another institution. And so a broadly engaged scientific team really includes all of the stakeholders, all of the potential stakeholders with an interest in the health need, in the research, in its implementation, in all the stages. Click, please. And so creating that broadly engaged scientific team where everyone is an authentic partner requires that everyone's ideas are granted the same respect and the same opportunity to evaluate their value and their feasibility in incorporating them into a program, a project, a protocol. I have heard not only investigators scoff at a community idea, but also community members sometimes scoff at an investigator's proposal for what they might need from the community. And so humility on all sides and respectful listening, because as mentioned earlier, everyone's an expert in something, and really a curiosity about why you would think we would need that, how would that be valuable, is sort of the core to building the trust and the real partnership that allows broadly engaged translation to occur. Click, please. So one of the exercises we did early on with our trainees, and which ultimately we came to require from all our investigators before their protocol goes to scientific review, before their protocol goes to IRB, is an articulation of the arc of their research, whether it's starting at T1 or T3, the arc from discovery to public health impact. And we constrain them to plain language and to 3 or 4 sentences. What's the problem? What's the research going to address? How will it ultimately affect public health impact? Click, please. And so articulating that arc, particularly for the early translational scientist, helps to crack open the silo and help them see or begin to see the connections between what they're doing and where it might ultimately go, and get them out of the silo. It can impact the way a T1, T2 assay development ultimately measures the biologic or clinical variable that's important in clinical application to make a difference. It can help in the planning to take advantage of the later translational existing initiatives to facilitate implementation and so forth. Click, please. And so it's also important in these broadly engaged teams to act, to think bidirectionally, not just in communicating and listening bidirectionally, but also to think about the trajectory, that arc from the bench to the bedside and back, because it's not linear moving across the translational spectrum, which is why you need to be prepared to think about the opportunities all along the spectrum. So a clinical need might drive a bench investigation that might lead to a molecule, that might lead to clinical trials. And in trials, the heterogeneous therapeutic response might then drive a question back to the bench. And so understanding the whole spectrum can add value to the research all along the spectrum. Next slide, please. So a very simple, maybe simplistic diagram, but to remind us that we need to bring everyone to the table across the translational spectrum, not just the basic scientist and the scientific collaborator, but also the clinician, the community clinician, maybe the academic clinician, the patient, the community members. Click, please. And other partners, the diagnostic lab that might be involved in the testing or the development of the assay, the agency, whether it's FDA or public health, the sponsors, et cetera, the advocacy groups and other representatives of community and rights groups, and the policy groups that ultimately may be impactful in whether the therapy is reimbursed and funded. And so when we first began designing our process for building these broadly engaged teams, full spectrum teams across the translational spectrum, we somewhat naively thought we would introduce the partners, facilitate the partnership, build capacity all around, and then step out of the way. Turns out it's not possible. It's a complex, organic, constantly changing set of relationships. And because of their distance initially across what I would call the translational divide, we found that it's extremely important to have a skilled navigator that continues to facilitate this partnership for the life of the project. We have projects we've been facilitating for 8 years, 10 years. And so that also means that institutions need to anticipate the resources, the expertise, and the support that's necessary to facilitate these teams. So we formalized this model in a paper we published back in 2017. And truth to tell, the diagram was a little cruder then. This is our more polished version of the diagram, but it centers on that collaboration building process at this in the beginning, the circles going around, and ideally all the partners enter at the building collaboration stage before the protocol's decided, before the aims are determined, before conduct's underway. But in truth, partners can be added at any time because it is organic and the details are negotiated constantly among the partners. And critical to the success of this community engaged navigation of the research process is the infrastructure that the partners bring. So in the upper left, CDN is Clinical Directors Network, Dr. Tobin's Practice-Based Research Network, and the infrastructure they bring that helps sustain the partnership includes experience and methods and skills in quality improvement, design of patient-centered outcomes and comparative effectiveness research, experience and resources for training community clinicians and communities, and other methods. What Rockefeller's the academic partner brings is expertise in laboratory investigation and mechanistic. In a process we have called translational research navigation, which is the protocol building, the actual writing of a GCP-ready protocol. We have trainees, MD, PhD, and MD scholars, and then other core resources like bioinformatics, biostatistics, and the ability to disseminate those research methods. So what's not on this diagram is the infrastructure that the community brings, that the community partners bring. We couldn't figure out how to draw it all. But together we've been able to conduct community engaged research, patient-centered outcomes research, all the types on the right across the spectrum of translational research. Next slide, please. This is a screenshot from our webpage where this is just one page of many projects that are listed. And what I wanted to bring to your attention is that we explicitly place our projects on the translational spectrum. So this is actually an interactive webpage, and you can click on the different phases and it'll bring up the different projects that incorporate and aim along that phase of the translational spectrum. You could see that not every project traverses the entire spectrum, but many hit more than one, often three, occasionally four phases of the translational spectrum. Next slide, please. So it takes time and trust, but something that isn't always discussed is alignment of incentives. And I call this my something for everyone. There has to be something for everyone in the project, in the protocol, to keep the partners aligned and to sustain the partnership. If there's nothing there for you, why would you stay? Next slide, please. And so those of you who join our breakout this afternoon will work with us in this matrix. Below the translational spectrum, we've included the partners, the priorities, hypothesis aim measures, and importantly, the outcomes. And going through this exercise for a hypothetical project or a real project around an unmet health need helps align the partners, helps them to see the opportunities, the differences, and how the outcomes that are meaningful to each of the partners at different stages of the translational spectrum are different and important. And with this information in advance, you can design intentionally, deliberately, to make sure that there are meaningful outcomes for all of the partners to help sustain this kind of relationship. And with that, I'd like to actually turn the podium over to my colleague, Dr. Tobin, who will describe to you in detail some of the projects that we've been able to develop and sustain across the translational research spectrum.
Thank you so much, Dr. Cost. And I'd like to thank Dr. Pachter, Dr. Carbon, Dr. Saunders for inviting us to spend the day with you, sharing some insights and reflections on the model that Rockefeller and CDN have been developing and disseminating, and to try to share with you this morning some concrete examples of how these partnerships have been developed over time, the kind of scientific questions that are generated by all of the stakeholders, and the kind of methods that are necessary for practical research in practice-based settings. And so I'll begin with acknowledging Dr. Angelet's comment about common values and how important it is to identify those common values, because oftentimes the stakeholders have very different reward systems and very different priorities. And so the underlying challenge is to try to identify those common priorities, those common reward systems, and at the same time to value and respect those differences and ensure that all of the stakeholders achieve and experience the benefits of this collaboration. So the other part of this discussion is about the work of a practice-based research network, or PBRN, Clinical Directors Network, and our N-squared PBRN is a network of safety net practice-based research networks that work with federally qualified health centers and other safety net practices across the United States, and have been building these observational and experimental studies that improve patient outcomes, have widespread utility within national and increasingly international clinical and public health domains that they address, and then with an overall focus on patient safety and on studies that align with practice and in no way ever compromise patient safety. If I could have the next slide, please. Let's go to the next slide. And so the N-squared PBRN has now 12 safety net practice-based research networks and 3 PCORNet clinical data research networks and a number of health information exchanges that are part of our network of networks. And through this, we've created an approach with our partners at Rockefeller that we refer to as full spectrum translational research. And if I can have the next slide, click on this, please. So it begins with basic science questions, and next, click again, please. And also focuses on public health impact, and next, click, by focusing on studies of both basic science, clinical practice, and health policy. And so we've tried to take this notion of the translational research spectrum and blue highways and extend it to include both basic science, health policy, and then really hold ourselves accountable to the question, are we improving public health impact? Does the work, does the outcome of the partnership enhance the health of communities? So I want to begin with one example that focused on a very specific scientific question. If we can go to the next slide, please. The next begins with a discussion of Dr. Alex Tomasz, who is a microbiologist at Rockefeller University, runs the Laboratory of Bacteriology, and really has developed, I think, a very critical understanding of the genetics of how bacteria develop antibiotic resistance. And this is an increasingly difficult and common problem, both clinically and in the research setting. And Dr. Tomasz established a New York area hospital surveillance network where he focused on studying the genetics of hospital-acquired methicillin-resistant Staph aureus, or HAMRSA, and found that the majority of them belong to very specific sequence, which he refers to as the New York-Japan clone. And when I met first with Dr. Tomasz, his interest was in trying to understand whether an emerging infectious disease referred to as community-acquired MRSA followed a similar pattern. So if we can click again, we took a look at those neighborhoods where he did his research, and we identified, indicated in the red boxes, a number of community health centers that were in similar neighborhoods or adjacent to the places where Dr. Tomasz had built his network and collected biological specimens. And this is actually a very early version of our collaboration with 6 federally qualified, or 6 community health centers in the New York metropolitan area that followed along with Dr. Tomasz's hospital-acquired network. And we put together, with the support of the CTSA pilot programs initially, and then with the support of a competitive administrative supplement, a surveillance network in these 6 locations with this very diverse group of investigators and collaborators to try to understand the process of diagnosing, treating, and preventing skin and soft tissue infections. And so the next part of this discussion is really to try to work through what were the questions and the interests of the different members of the team, and how were we able to support their expression in systematic research. Next slide, please. So the first one that I want to show is really a partnership between, on the right, a physician assistant, Scott Salvati, who worked in one of the health centers in the Bronx, and a molecular epidemiologist postdoc, Maria Pardos, who worked in the Tomasz lab. And we began by understanding the clinical presentation and at the same time understanding the biological substrate, the genetic differences, the variations in MRSA. And this was recognized at the National North American Primary Care Association as a model approach to conducting full spectrum translational research that brought together CTSAs and practice-based research networks. So this conversation, if I can go to the next slide, continued with Dr. Pardos's work in understanding through pulse field gel electrophoresis, next click please, that in fact the clonal types in the community-acquired MRSA were not the same as those that Dr. Tomasz had previously found when he studied the hospital-acquired networks, hospital-acquired specimens. And so it was really critical to provide an opportunity for the Tomasz lab to continue their work while learning more about the partnerships, more about the clinicians that were treating skin and soft tissue infections in the community setting, and understand what some of the patient concerns are, as you'll see in a moment. We go to the next slide, please. So at the other end of the spectrum, click again, please, we have a nurse practitioner who works with us, Nancy Jenks, in Westchester. And Nancy sees a very diverse population of immigrants from Central and South America, and was quite interested in understanding the pattern of antibiotic resistance among the immigrant population that she was taking care of. Click again, please. And so she presented a paper at an international conference to the International Society of Travel Medicine. Click again. And we published this paper together in the journal from that organization. And most interestingly, we approached this from the standpoint of assuming that because antibiotics are typically available over the counter in many of the countries that Nancy was interested in understanding better healthcare and patterns, we actually found the opposite, that in fact, most of the non-US born participants in our initial observational study didn't have MRSA, methicillin-resistant Staph aureus. Their skin and soft tissue infections were actually caused by methicillin-sensitive Staph aureus. And so we dug a little bit deeper into their lifetime exposures to antibiotics and actually determined from this experience that these patients had a much lower lifetime exposure to antibiotics, which may have contributed to the susceptibility during these skin and soft tissue infections. We go to the next slide, please. In the process of discussing how we could further disseminate the results of this study, another nurse, Rhonda Burgess, mentioned a partner that she worked with, click again, please, who ran and two more clicks, who ran a network of barbershops in East Harlem. And Dennis Mitchell has a network called Denny Mo's, and we went to talk to him because he's a leader in his community, a business leader with an interest and a commitment to public health. And when we talked about skin and soft tissue infections and MRSA, because Rhonda Burgess, the nurse indicated at the left, mentioned his role as a community leader, he was very, very interested. He had never heard about MRSA. When we showed him some pictures of what that looks like, he reacted that he sees this almost every day and was concerned, could he catch it? Could his other workers in the barbershop catch it? Could they transmit it to their family and household members? Could they transmit it to their other clients? And this led us to develop a continuing education program on prevention, on infection prevention and prevention of infection and transmission. that was targeted towards barber shops and other aesthetic care workers. And as you can see from the map on the left, when you look in one single area surrounding one of our partnering community health centers, we found that there are really an extensive number of barber shops, beauty salons, nail salons, makeup emporiums that serve the same populations that the health centers serve, and often serve populations that don't necessarily seek a routine source of health care. So we, as others, have developed strong partnerships with the barbershops, developed a continuing education program that we then redeployed in a subsequent study. Can we go to the next slide, please? So in this particular conversation that began between Dr. Shirish Balachandra, one of the primary care clinicians in one of our Bronx health centers, and Dr. Maria Pardos, the postdoc in the Tomasz lab, at every one of our monthly in-person meetings, we would invite one of the clinicians to present a case study of a patient presenting with skin and soft tissue infection that they enrolled in the study and treated. And we'd also ask one of the laboratory investigators to present a paper that had recently been published in the literature related to the topic of interest, and asked to really create this conversation about the clinical and laboratory basis of our shared investigation. And in one particular session, Dr. Balachandra mentioned a patient that he had presented previously who now reappeared at his health center with a recurrent infection, in spite of the fact that he had applied incision and drainage previously and had targeted his antibiotic treatment to the antibiogram. Yet she kept coming back with a recurrent infection. And this actually led to a question when Dr. Pardos looked in detail at what the molecular characteristics of this particular pathogen were. We can go on to the next slide. And again, created this dialogue between the clinical, my patient's MRSA keeps coming back despite treatment, and now her sister who is visiting her has it too, and she's very upset with me. And so Dr. Pardos was interested in understanding, if we click on the next slide, whether the clones responsible for the recurrent SSTI were similar or different. And click one more time, please. And so she had the opportunity to determine that, in fact, this was the same pathogen that was infecting not only the wound culture, but also some of the surveillance cultures. And so when we had this set of presentations at that day, if we go to the next slide, there was almost this incredible eruption of interest and conversations that were going on, that the patients were interested in understanding what was giving rise to recurrence and what could be done to prevent recurrence, that the clinicians were really concerned about whether incision and drainage and antibiotic treatment was the right approach. The laboratory investigators were interested in understanding whether this phenotype reflected a single or multiple genotypes. And we had noted that about 1 out of 3 of the patients enrolled in the observational study had recurrence of either their MRSA or MSSA. So it was clear from this conversation that our attention needed to be focused on understanding the predictors and the prevention strategies to prevent recurrence and household transmission. Next slide, please. So we prepared another grant that went to PCORI, the Patient-Centered Outcomes Research, and I want to try to use this to demonstrate the point that we work through each of the stakeholders' specific interests to develop an effectiveness and implementation aim, where we work with promotores to deliver a home-based intervention that has been shown to be effective at preventing infection transmission in an ICU setting, but had never been deployed in the community or in a home-based setting, and had never been deployed with community health workers or promotores. So this was the primary comparative effectiveness aim. Next slide, next click. At the same time, we were interested in understanding whether there were patient-level factors that were influenced by the intervention, such as knowledge, self-efficacy, their decision-making autonomy, that were associated with reductions in transmission and prevention of recurrence. Next slide. Next click. We also had a mechanistic and molecular aim to try to understand the intervention between the bacterial genotypic and phenotypic variables on the decontamination, decolonization, SSTI recurrence, and household transmission. And then finally, we had this interest in understanding the evolution of the team over the course of now what was about 5 years worth of collaboration. Next slide, please. So time won't permit me to go into this, and what we did find in this second clinical trial, click again please, was that the intervention was not successful. As you can see here, we had about 11% recurrence in the electronic health record between the experimental and the control group. So in spite of our best efforts, we were not able to significantly reduce the incidence of recurrence, although this 11% rate was about a third of what we had previously observed. Next slide, please. But however, click again, please. We did demonstrate reductions in the secondary outcomes, decolonization, as a result of the intervention. So if you click on the next slide, please. This was a very large team of collaborators, including patients, clinicians, health services researchers, laboratory investigators, a number of private companies, several academic institutions, and a number of scientific advisors across the United States. And we're grateful to both NCATS and PCORI, as well as to AHRQ, for providing some of the resources that made this study and this ongoing partnership, which continues to this day, possible. And I'll just mention, the governor mentioned the outbreak of COVID among poultry workers. We're currently partnering with the U.S. Department of Agriculture, which has interest in understanding the degree to which some of MRSA may be transmitted through poultry. And so there's another natural alignment here. Next slide, please. I want to briefly talk about a second collaboration, which followed a somewhat different model, but this is working with a senior center, Carter Burden Network, that provides a variety of food-related programs, health and nutrition programs, and other services to seniors who are aging in place in the community in East Harlem, in the Upper East Side of New York, and in other parts of New York City. Next slide, please. So in a very preliminary pilot study supported through our CTSA, we learned that many of the participants at Carter Burden had hypertension, and many of them, when they presented for health screening, had poorly controlled blood pressure. And so it was clear that improving the cardiovascular health of this population was a priority for the participants at Carter Burden and for the investigators. So we developed this collaborative study to test whether we could implement the dietary approach to stop hypertension, or DASH diet, in the congregate meals that were provided by Carter Burden. Next slide. So we received some funding, and this is a very diverse research team that continues to work with us on implementing this and the future studies that are being developed as we speak. Next slide. So in this, we were interested in determining whether we could implement DASH diet through the congregate meal programs with additional education and behavioral support, and whether that in turn could lower blood pressure in seniors who undergo the program. And we started this in 2 different settings. Next slide. With support from the Administration for Community Living, and participants received their meals at the centers that were now aligned with the DASH diet. They attended a series of health and nutrition education sessions. They had their blood pressure measured on site by a third party, and they all received an Omron 10 blood pressure device and training for home blood pressure monitoring and recording of their blood pressure. Next slide. So the intervention was focused on the change in mean systolic blood pressure at 1 month and the increase in proportion of blood pressure control according to the JNC 8 guidelines. We measured blood pressure, pulse, height, weight, home blood pressure monitoring, and we conducted a series of different surveys and really tried to ensure that the dietary changes were palatable and acceptable. So there was a lot of interaction among the participants, the staff at Carter Burden, particularly the staff who were responsible for preparing meals, as well as the Department for the Aging in New York City, the agency that has to approve any changes to the diets to ensure that nutritional completeness of meals provided in these congregate meal settings. Next slide, please. So to just fast forward, then there was COVID, and we had to dramatically change the way in which meals were delivered and blood pressure was measured. Nevertheless, we did observe that we had a 4 millimeter reduction overall and an 8 millimeter at one of the two sites. These are both statistically and clinically significant reductions in blood pressure. We also found an increase in the proportion of participants whose blood pressure was well controlled by JNC-8 standards. So we determined that it is indeed possible to implement the DASH diet in congregate meal settings. Next slide. So again, this demonstrated the statistical and clinical significance of the reductions in blood pressure and the sustainability of that over 5 to 6 months. And so this makes us very, very confident that this is a program that really should be disseminated and implemented much more broadly. Next slide, please. So we determined it's feasible to implement these DASH-aligned meals, that we could accomplish both the implementation and the clinical effectiveness. We learned a lot from the process about working with the New York City Department for the Aging and adapting the meals. We developed and initiated a partnership with the New York City Department of Health and the Department of Aging, and are looking to expand this among a broader range of senior centers that provide meals and other health and educational services to the populations of older adults who are aging in place. Next slide. So it showed that this is innovative, we could do this, and despite COVID's interruptions in the way meals were delivered and our blood pressure was measured, we still demonstrated the clinically significant reductions, and that at least a third of participants continued to monitor their blood pressure at home with the devices that we provided and the training that they received. And we noted that women and Black participants were much more likely to continue using their home blood pressure monitoring. Next slide. I'll mention very quickly that in parallel with the community-based study, Dr. Costa and I have been training a clinical scholar at Rockefeller who is a nephrologist. Dr. Bielopolsky is interested in understanding the underlying mechanisms that contribute to the success of the DASH diet, and developed an approach by admitting 9 community members to the Rockefeller University Hospital for 10 days, where we could control their dietary intake and feed them a DASH diet, measure their output in terms of urinary sodium and potassium, provide ambulatory blood pressure monitors so we could get a much more ongoing source of variability of their blood pressure, and really try to understand not only does the DASH diet work, but how does the DASH diet work? So this presents another model of creating this full spectrum partnership that spans the basic science through the community. Next slide, please. So I want to just wrap up by acknowledging the partnerships at Carter Burden, at Vital Care, the Department for the Aging, and an advisory committee consisting of leaders of a number of community and governmental agencies that provide care for older adults in New York City, and really indicate that, again, this is an illustration of how these full spectrum partnerships can provide insights both clinically, epidemiologically, and mechanistically. Next slide. So to sum up, this approach really takes the idea of tightly controlled efficacy studies that are conducted in academic centers and brings them into community-based settings of effectiveness and implementation. Click again, please, where we try to embed mechanistic studies, click again, and studies that look at implementation, scale up, and sustainability. One more click. So here we try to simultaneously study effectiveness, implementation, and mechanisms to understand what works for which subgroups, and at the same time, how does it work with the underlying basic science mechanism. Next slide. So one of the principles, click again, that always is, I think, behind every one of our studies is how to combine research that looks at effectiveness and implementation as part of this design. Click again. And these are typically pragmatic studies that follow this concept of real-world evidence in real-world settings. And if we click to the next slide, and just click several times, because we have, I think in synopsis, the full translational research spectrum illustrated in, keep going, in Camp 1 and Camp 2, the community-acquired MRSA project, where as we think about the publications that have come out of this study, keep going, they really have helped us by bringing together all of these unique interests and unique expertise. They've allowed us to explore simultaneously questions of relevance at the full spectrum. And if we go to the next slide, and we're in the process of doing a similar approach with DASH, looking both at the underlying basic science electrolyte mechanisms in the kidney, all the way through an intended and planned multi-agency dissemination and implementation pragmatic cluster randomized trial. Next slide. So to try to bring this together, as Dr. Koss mentioned, click again. Oftentimes the basic science and public health questions align, if we click again, by looking at these environmental policies that contribute to well-being, and the basic science questions that can explain public health impact, then click one more time, often take us back to understanding some of the heterogeneity and diversity. So if we go to the next slide, the shared goal is to embed the full spectrum of translational research into practice, or translating practice into research. And that involves principles of engaging all members of the team, designing programs that can be disseminated. The model that we've developed at Clinical Directors Network focuses on clinician engagement and clinical leadership. And with our work through PCORI, we've developed an approach to engaging patients and the leadership of patients and patient advocacy groups as integral partners in the research design, all of which have helped to build this science-based ecosystem of building sustainable bridges between practice and research. And in my final slide, I just want to mention that as part of our current CTSA grant, we're interested in building, in collaboration with other partners such as ACCEL, how do we build these full spectrum translational research teams across multiple institutions so that we can disseminate through collaboration this model of full spectrum translational research. And with that, thank you so much for your attention.
That was wonderful. Thank you so much, Drs. Costa and Tobin. If anybody in the audience have questions, please feel free to put them in the chat. We'll try to answer as many as possible in the time that we have. And for those questions that we don't have time for, we will close the loop and get the answers to you after the conference. So again, thank you. We do have a few questions to get started. I noticed in one of your initial slides, you had mentioned navigators, and many of us are used to use of community navigators in clinical care and clinical medicine. Can you give us some examples of use of navigators in clinical and translational research?
Well, actually, the navigators, Rockefeller has a fairly small team, and as you know, community engagement is often under-resourced. And Dr. Tobin and I and our community engagement specialist, who will be helping with the breakouts this afternoon, Anuradha Hashemi, we are the navigators. We facilitate, we have the knowledge to translate the culture and the language and the help align the aims. And so we facilitate the meetings between the many stakeholders, and we aim to develop others within the group as the navigators. Jonathan, maybe you'd like to add to that, but it's the ability to understand the full spectrum, to understand how misunderstandings can happen and smooth them over, and support that respectful curiosity that keeps everyone collaborating. Jonathan?
Let me take on the next question, because I think it addresses this, which is the question of sustainability. And the short answer is sustainability is one of the most critical outcomes to address in terms of implementation research. One of the challenges in funded research is that oftentimes the timeline, a 2-year or 5-year grant, doesn't really permit a full assessments of sustainability. However, there are two components that I think are important to consider. One is, in all of our studies, we've put together a measurement model that allows the possibility of continuing to collect information in the populations that have been part of the study, also potentially allowing us to expand those computable phenotypes to other partners and to accelerate their engagement in research. And so that's one part of it, having the measurement system developed and refined during the research study. The second component goes back to the research team. And so having that team continue to work together on the next project and the next project is another mechanism that helps with developing sustainability. And Navigators are part of that team and potentially part of the glue that can continue to keep that team together. So I think this is a really critical question, one that deserves a more thoughtful answer, but at the same time, I think the short answer is yes, sustainability has to be part of it, and these are some of the mechanisms and methods that can help us continue to address sustainability.
Thank you. We have another question. Sometimes T0 and T1 scientists don't necessarily immediately see the need to engage communities in research. Have you noticed that? And if so, what advice do you have to overcome this?
Well,
so let me address that one, because the conversations that I have typically begin with, can't you just send me the specimens? And I've heard that with the same words or similar words in almost every discussion with investigators, not only at Rockefeller, but other basic science investigators. And so the answer is, well, we could potentially do that, but we're not going to, because that's not the model of full spectrum community engaged research. And so some investigators are simply not going to be interested in that. And one way to perhaps get them more interested is to work through members of their team, members of their lab. So in some instances, we work through their clinical scholars, their postdocs, or other members of their team who may be more open to the idea. But I can say that most of the investigators who have taken that approach have benefited from coming to these monthly meetings, from giving up a little bit of their time, but expanding their perspective. So I think we just have to ask, we have to be welcoming, the teams form over time, and be prepared to say to them, we'll send you the invitation and hope that you'll join us when it's convenient for you. Rhonda?
Yep, I agree, but I'd like to add to that, community engaged research isn't for everyone, and the incentives aren't yet aligned. And so the degree to which leadership and funding agencies value and even demand community engagement goes a long way toward incentivizing basic scientists to participate. The other aspect is that you don't have to join the full spectrum right away. Learning can be incremental, and community engagement can be incremental, and helping a basic scientist who really is working just on a mechanism, on a pathway, to understand how giving a full spectrum translational town hall, which I'll take a moment to describe, doing that and engaging the community and just sharing what you're doing is, opens the door. And so it might not be this project or protocol, it might be the next one. It might be incremental for that investigator. And so a full spectrum town hall is a public presentation that incorporates some basic science, some clinical science, some patient advocacy, some policy, and has not only all that spectrum of presenters, but that spectrum of audience. And we found it to be helpful in advancing partnerships and relationships. And it's not a recruitment opportunity that now I've shown you the spectrum, can you join my study? It's a partnership development activity.
That's great. I'll emphasize the importance of funders in knowing about community engaged research. As an example, ACCEL, all ACCEL pilot proposals need to have a community engagement statement, whether they're a community engaged research project or not. So that's a great example of how funders can really influence the knowledge and importance of community engagement. Question, we often hear that research takes too long. How do you balance the time needed to conduct research with the community and clinical needs for immediate answers and outcomes?
Jonathan, I'll give you that one.
So I think what's really critical is to develop programs that provide resources to our community partners and that are identified by the partners. And in fact, that's what we hope to model in this afternoon's workshop, is how do we bring together the resources of academic medical centers, academic research institutions in the service of communities. So I think part of it is going back to understanding that the priorities are different. And what we try to share with our community partners is that if this partnership is going to be successful and going to be maintained, the partners have to understand the timeline and accept and respect that it does take time. When we submit an NIH grant, it could be, if we're lucky, a year between submission and startup, if we're lucky. And if it's a 5-year grant, it might take us another year to put the primary effects into publication. So we're saying to our partners, this may take 7 years to give you the answer. Well, people aren't going to have 7 years to wait. So we have to have designs that provide benefit in the shorter term. They may not provide the concrete answer, but they might provide useful resources, useful education, and clinical support that make the participation valuable in the shorter term.
Great. We have a few questions from our community members. One is, are you aware of a situation where a community person is listed as the co-PI or co-I on the research project? Is this a way to break down the mistrust between researchers and communities? Is this possible?
So I'd like to say, first off, for the ACCEL Nutrition Innovation Grant, the NIH funded grant for 2 years, our community partner was the PI of the grant. So they were delighted. This was their first NIH grant. There was certainly a lot of co-design and co-writing involved, but she was in charge of the purse. She was the PI, and she was a co-PI on the clinical protocol for operationalizing the project. So there's one.
Yeah, and in the MRSA project, is those of you who've had experience with PCORI may know that it's a non-starter if you can't demonstrate it. When you write the grant, you talk about patient engagement in the future tense, it's unlikely to get funded. So it's a reminder that the funder really can directly influence, and PCORI has had that direct influence in a relatively short period of time on our work and the work of many other teams, where patients are listed and included as investigators. And I hope that the second example of the MRSA intervention trial demonstrated the importance of that level of engagement.
So it is a demonstration of actions, not just words, that the community partners are co-investigators and co-authors and often presenters. So for the MRSA project, the Camp One project, the first paper was written by a community clinician, right? Dr. Balachandra's paper was the first one, Jonathan, wasn't it?
Yeah.
And
so
go ahead.
So it's important to give each voice an opportunity to articulate a scientific question, and then take each of those questions as critical and valid, whether it came from the barbershop experience, or it came from the clinical experience, or it came from the laboratory experience. And so I think it really is a reminder that that kind of commitment takes time and effort, and reflects not only the diversity of the experience and expertise of the team members, but it really does reflect the diversity of the needs of the different participants. And those of you who've worked with federally qualified health centers know that in order to receive HRSA funding, Health Resources and Services Administration, you have to be led by a community board that consists at least of 51% of residents and users of the federally qualified health center. And that's a way of ensuring community accountability. And it's also a way of diversifying both the leadership and consequently the workforce and the patients who are served by those health centers. So FQHCs, I think by their genetics, have a design built in to ensure community accountability, community representativeness.
Definitely.
When we were developing, just one more comment, we were developing our model, one of the measures of the effectiveness of our community engaged research navigation process was whether those community aims made it into the protocol. So fine and good to have the meeting articulate everyone's goals and aims, but did those aims make it into the protocol? And that is the mapping on the translational spectrum that you see in the paper and that you see on our website. Those are not retrospective, those are the aims of the protocols.
And that's a good example of engaging communities early on in the research project. We're often considering, how do we communicate results as quote unquote community engagement, but really community engagement needs to start at the beginning of a project, at the conception of the project. So I really appreciate that. We have a few questions about the DASH project. First of all, it was really interesting that as you conceptualize projects, you put them on the translational spectrum, and many projects have multiple different T levels, which I thought was really interesting. And I think that the DASH was one that had from T0 to T2 or 3 or 4. Can you explain the different translational components of that project, number one? And then secondly, a question was asked, was the DASH diet continued at home or just at the senior centers? And what communities were the senior centers located in?
So I'd like to answer the second question first and then give Jonathan the first question. So we knew that we wouldn't be providing 100% DASH. The goal was not to feed people an around-the-clock DASH diet and see if it lowered blood pressure. We already know from efficacy trials that an around-the-clock DASH diet in a controlled environment lowers blood pressure. We wanted to model the DASH diet by working with them for palatable menu changes and by providing it there and providing social and educational and behavioral support for changing nutrition. And the hope was that by modeling that, they would continue those DASH principles at home. There is no good measure that's actually a feasible measure of DASH compliance. All of the DASH adherence surveys that we found were like 35 pages long, not possible to implement. And so, it was pragmatic to an extent. We provide it. We provide all the educational, social, and blood pressure monitoring support for self-efficacy around this, and we see what happens. They were also the communities were in East Harlem and a section of the Upper East Side. The Upper East Side you traditionally don't think of as underserved, but many of the luncheon club attendees in the Upper East Side are actually homeless. Jonathan? Sorry, housing insecurity.
So I think the critical issue here was how could we work with existing structures in community-based settings that are this gets back to the question of sustainability, so that if we were to introduce a dietary intervention program that ran for the 2 years of the funded research project, at the end of the study, we would publish our results in peer-reviewed journals, give presentations, and then leave it to policymakers and advocates to change the food environment to make it more DASH compatible. We view full spectrum as having the responsibility to continue and to engage in that discussion, to bring the partnerships and the stakeholders together to try to make those policy changes, and to continue to work with communities and with community participants so that they, in fact, are the messengers and that they are the ones who provide the ongoing sustainability of the importance of self-monitoring of blood pressure at home and of changing the approach to nutrition and eating that aligns with principles and practices that have been shown to be effective. So what's really unique here was the focus on implementing an effective set of evidence-based strategies. And we have no shortage of really good scientific evidence of how to reduce cardiovascular risk in underserved and diverse communities. What we really lack is good evidence on what are the best implementation strategies, and are they community health workers? In some instances, that's the right approach. Are they farmers markets, food pantries? All of these have an important role to play, so that the kind of studies that we want to design might compare head-to-head a series of different strategies and try to understand which ones are most implementable in which communities, which ones have the sustainable clinical benefit, and which ones can be sustained within those communities without additional external resources. So this gets back to the concept of pragmatic research and engaging partners who are in the business of delivering those services, whether they're social services or healthcare services, and helping them to test in their settings their ability to implement and sustain these effective strategies.
So I could address the translational spectrum briefly if we still have a minute for that. So we started with this T3, T4 idea, implementation of the evidence-based intervention in this community environment. And as Jonathan mentioned, the full T4 policy comes, with the next project or projects where we've engaged all these other partners and need actually to figure out how to make it broader and sustainable across many centers. But we wanted to understand the DASH diet definitely lowers blood pressure. There are a lot of hypotheses about how it might work. It's probably multifactorial, but it's not well understood, and it probably doesn't work for everyone. So we wanted to embed a mechanistic piece, a T2 piece, to understand how it works. But we found that in the environment of serving meals in a senior center, it was not going to be possible to collect specimens. And so with our clinical scholar, Dr. Dana Bielpowski, we actually did a parallel study, fully integrated in the thinking, but in fact, a separate study, an inpatient study at Rockefeller University. And now we didn't need another effectiveness in patients efficacy study because we already know that, but she had to replicate an efficacy study to enroll people with mild hypertension, no known renal disease, in a 2-week hospitalization where she gave them a highly controlled diet. And what two of the goals during this project, there was a T2 goal, which was to develop a better clinical measure of DASH adherence, since we can't do that survey, that 35-page survey. And typically what's used for DASH adherence is a 24-hour urine collection, which is also difficult. So she ended up validating that a morning spot urine is as good as a 24-hour urine collection in monitoring DASH adherence because of the flip in electrolytes that happens. You usually have high sodium, low potassium when you're eating a Western diet. When you're adhering to the DASH diet, it flips and you have high potassium, low sodium in your spot urine. So that was a T2, assay development, sort of T2, T3. But she also was interested in exploring a hypothesis about what actually happens in the kidney renal tubule. And so her discoveries are still in analysis and not yet published, so I'm not going to give too much away. But what she did was explore a new approach to a liquid biopsy of the kidney to understand what's happening on the renal tubule before, during, and after DASH. And she has some interesting preliminary data that points to support of a hypothesis that she had proposed about what's actually happening in the renal tubule. And that's a very basic physiologic mechanism. So we would call that T021.
Thank you. We have other questions, but I think we're about time for a break. I want to thank you both for a great discussion, a great talk. We look forward to continuing it at one of the workshops this afternoon. So thank you, everybody.